Okay, now let's see some details. So NGS data, to give some definitions and describe a bit the process. In order to know during the class what I will talk about. So let's say that we have a tissue here. This is an organism. So this is a tissue of an organism, and of course it has different cells. So these are cells. And just to make things simpler, every cell has some piece of DNA. So this is which ideally it should be exactly the same for all cells, but of course you know that you have some mutations from cell to cell, so it's possible that it's not exactly the same. If I remember well, you have one mutation every million of base pairs, so two molecules here, two DNA molecules, might have a difference every single every 1 million base pairs, but this is not important. So let's assume for the moment that all of them are exactly the same. Okay, then what you do is that, of course, you extract all this DNA. So here you have whatever it is, Eppendorf or whatever it is, of all these molecules, which should be exactly the same. And then you sonicate it. Sonication. So with sonication, what will happen is that this piece of DNA will just become very, very small fragments. Okay, let's draw it better maybe. Just break everything, in small fragments. So all these molecules broke in pieces like that. Okay, now what is important to understand at first step here is that this piece of DNA, you actually have it here multiple times, because you have it from this molecule, and you have it from this molecule, and from this molecule, and from this molecule, and so on. So let's say that these are these pieces of DNA here. We should be exactly the same. It should, well, no, part of it should be exactly the same. And the other thing to understand here is that these pieces of DNA that corresponds here to the left end of the chromosome, they don't have exactly the same length. So if I will magnify this image here, let's put some numbers here, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Then perhaps this piece is this one. And the piece corresponds to 2 is this one, and the piece corresponds to 3 is this one, and the piece corresponds to 4 is this one, and so on. They don't have exactly the same length. Why? Because of sonication. I just write it here. So sonication means that you put ultrasounds to break the DNA, and because breakage is, it's a stochastic process, some DNA might break here, and the other DNA might break a bit further. Okay, Now these pieces, so this is step number 0, let's say. Step number 1, we make a size selection. Okay, let's use blue again. In size selection, we don't really want to keep very short pieces and very long pieces. So I don't remember exactly the sizes, but this should be, I don't know, around 300 base pairs. If I remember well, it's between 200 and 600, so on, but it doesn't really matter. Perhaps it's changing, it changes during time, also it depends on the technologies, and perhaps in current Illumina it's also bigger. Let's say around 500, doesn't really matter. Which means that you will discard some of them. Let's say we discard this one, and let's put one more here. And we have all these guys. Now step number 1 then, so step number 1 size selection, step number 2 is the following. We have all these pieces of DNA that we have kept, which are called now fragments. So these are the fragments. And what will happen in Illumina is that so in Illumina, okay, I must say that now, in Illumina we have the single and reading and the paired and reading. Okay, let's see what will happen now. Let me take one piece of this. Let's say to take this fragment. So fragment number 1. Fragment. It's this one. And let's say that this is 523 base pairs. With Illumina we will not read all of it, but what we will read here, after putting here some primers or ligands or whatever it is, what we will read here, let's use this orange color or yellow color maybe. Yellow is not good. Green. Depending on the technology that I will tell you a few things about, we read a certain amount of base pairs on the left end of the fragment. So some initial technology a few years ago was reading exactly 100 base pairs. So synthesis here goes all the time from 5 to 3' end and was reading exactly this piece of DNA, exactly 100 base pairs. If it reads only from one end, then this technology is a single end reading. But it is possible also that we have the complementary of it. So this is the reverse complement of the first one. Reverse complement. You will read also from the other side. So you will get also this information. Now, what you should pay attention here is that you will get 100 base pairs here. Yeah, let's use purple. 100 base pairs here. You have get 100 base pairs here, but perhaps you will never get this middle part, because this is 523 base pairs. So roughly you will get 100 base pairs on the left, 100 base pairs on the right, and then you will have something like 300 base pairs that you will never read for this fragment. But you will see later on that this is not a problem. Okay, so just to say everything from the beginning, to have in a more abstract way, if you have the fragment that is 523 base pairs, or some number of base pairs here, doesn't really matter, then you will get some information from here, and possibly, if you have paired end, some information from here. So this is paired end. You might have also single end reading, which means that you will get only from one side. This is 100 base pairs, this 100 base pairs, this 100 base pairs. And again, it means here that this piece of DNA you haven't read it for this fragment, and this piece of DNA you have not read it for this fragment. Okay, but then how you will get information for the whole genome? Now, in order to understand this, you must really go back here and remember that what you had in the beginning was in this mixture here you have several DNA molecules from different cells, and each one didn't break exactly at the same location. So of course for this fragment you will not read this, but it's possible that for another fragment, so this is fragment number 1, but if we go here and we have fragment number 2, which again corresponds to the same part of the chromosome. So fragment number 2 is this one, but it's a bit smaller. Okay, so fragment number 2 is not 500 base pairs, but it's, let's say, 300 base pairs. So again you will read this part, but now you will read also this part, and you will just leave 100 base pairs here. Which means that you will have from this fragment a read between from position 200 to 300. So in this piece of DNA that you didn't read, actually 200 starts from here and 300, let's say, is here. So you will get this information that you didn't get in the previous fragment, because you will read this one. And then you will have another fragment of DNA, for example. Here you have a break, let's say, in position 50. So you will read something that is between 50 and 150. So you will get something in this 50 and 150, in this area. So if this is 50 here, this will be 150 here. For another fragment, for fragment 3. So fragment 3 is a piece of DNA, let's say 300 base pairs or 320 base pairs, that didn't start from position 1 of the genome, but starts at position 50 and goes okay. So this piece of DNA starts at position 50 of the chromosome, but goes up to 370, just because its length we said that is through 320. So if we read again 100 base pairs here, then we'll go up to position 150 of the chromosome that we hadn't managed to go with the previous fragment. So if we try to put all of that in the context of the chromosome, let's go here. It means that if we have, let's say, human chromosome 1, that starts position 1 here, then what I said before is that we have managed to read from position 1 to 100, and this is because of fragment 1. This is not fragment 1, but it's due to fragment 1. So this piece that we read belongs to fragment 1. You remember fragment 1 was 527 base pairs, but we read just 100 here and 100 here. And because we read this one, it means that we read, so this is, if this is 525, this is position 527, it means that we read also, because of fragment 1, the 427 to 527. So this is again from fragment 1. And again its length is 100 base pairs. And then from fragment 2, we read again this piece, so this is from fragment 2, but we read a piece of DNA somewhere here, just because fragment 2 was shorter. So if I remember well, fragment 2 was 300 base pairs, so we read also, okay, I must make this. So we read also this piece of DNA from position 200 to 300. And then from fragment 3 we read from 50 to 150, I said, so this piece of DNA here is from fragment 3, and so on. So if we put all together, if we have human chromosome 1, we have read something like that. And so on. Maybe this should go. Okay, so if we put all together, we have read this information just because of the 3 fragments that we have seen up to now. Now if we would have millions of fragments, or several thousands of fragments, actually millions we have, then the image would be like that. and so on. Now, just to be clear here, this is not a fragment, it's a piece of a fragment. And these pieces of fragments have the famous name reads. So these are the reads. And we have, so in NGS data, typically we have several millions of reads. And what is important to understand is that, and to know that, these millions of reads do not come from the same molecule of DNA, but they come in fact from different, ideally exactly the same molecules of DNA. Because you remember here that we had DNA from a multitude of cells. Okay, so these are reads. And the process, so the computational process that we will see during the class of saying where a read belongs to. So what is the assignment between a read and the genome? So for example, say that this read is a read that corresponds to position 500 to 600. This process is called mapping. And one more thing that I should mention here, in the mapping in the mapping process, what I do, so when we do this mapping with tools, I will say some more things about that, but mapping is to assign a read to a specific location of the genome. Of course, you understand here that in order to do the mapping, I must have the genome. So for human genome, this is easy because we have what we call a reference genome here. Which is a genome that we created about, first time we created something like 20 years ago or 15 years ago, and then we started to make it better and better. I will say also a few things about that during the class, maybe in a different lecture. But we have a reference genome, and then we can map reads on this reference genome. We can do exactly this mapping process that I described earlier. Sometimes for organisms, other organisms that are not model organisms, we don't have the reference genome. And what we have there is just a bag of reads. These are reads that they are sequenced. We know the sequence of these reads. And what we want to do in a first step that we will not actually cover during this class will be to find these overlaps here, because these pieces of reads should be the same, and this read should be the same with this read in this part. So there are algorithms that try to, from this bag of reads, to put them in an order, find the overlapped parts and do something like that, and from this build what's called an assembly, which will be something like a reference genome. So for example, depending on what information these reads tell about this position, we'll recover that this position has a C here, because this read has a C, this read has a C, and this read has a C. So here we have a C in the assembly. Okay, I mentioned that because just to give some terminology here. So for us at the moment, the important thing is what is a fragment, what is a read, what is a single and read, and what is a paired and read. And what is mapping, which is the process of assigning reads to the locations, the positions of a genome, which we call a reference genome. Okay, and now we can, in the next video, we will see how these reads look like. Just to mention before going to the next video that I said before that some years ago Illumina had pieces of DNA that was 100 base pairs. This depends on the technology and the sequencer that we use. I think now we have 200 base pair reads or 250 base pair reads. This depends on the machine of the Illumina company, of the company. Okay, that's it for the moment.